some problem regarding to hormones. One of the hormones that can affect the hair growth is the thyroid hormones. So in a case of hypothyroidism, we have a hair loss. And also, as I mentioned, we have some problem in infertility, and actually reproductive system are affected by the thyroid hormones. You should know that if you have untreated hypothyroidism during the pregnancy, it can lead to the delay in the growth of the fetus. So this case known as a cretinism. Okay, so maybe I ask you for your final exam, because you will have 3 MCQ tests for each practical session. Okay, maybe for example one of the questions is that what is cretinism? Okay, you should know that this is a kind of hypothyroidism, and especially during the pregnancy. And sometimes the low concentration of the thyroid hormones is as a result of the lack of iodine in a diet. Because as I mentioned, some of actually you were absent, this is the structure of the thyroid hormones by the addition of the, this is the amino acid tyrosine, by addition of 1, 2, 3, we have a T3 hormone, and if you add another iodine, you have a T4 hormones. Whenever you didn't receive enough iodine in your diet, so you cannot produce the T3 and T4 hormones. These are the hormones that's created by the thyroid gland. Okay? So whenever you didn't receive enough amount of iodine, so it can result in a hypothyroidism. And as I mentioned, one of the autoimmune conditions is a Hashimoto disease. And most of the case of this hypothyroidism can be treated by the oral administration of the synthetic thyroid hormone, as maybe one of you or maybe one of the members of your family have this problem, and the physician prescribed levothyroxine. Okay, levothyroxine is a synthetic form of the thyroid hormones. So you can control it. About the goiter, this is actually enlargement of the thyroid gland, and it is very visible. Okay, the size of the neck actually increases, and it's very clear. This is a constant stimulation of the thyroid to produce more and more hormone, because the concentration of the thyroid hormone is low. One part of the brain, in the next slide I will show you, send the signal to the thyroid to produce more. So this continuous stimulation can result in increase in the size of thyroid glands. And hypothyroidism is one of the most common cause of goiter. So you should know that goiter is one of the kind of hypothyroidism. About hyperthyroidism, as the name implies, we have a high activity of the thyroid gland. So about the symptoms, they are completely in contrast to the hypo actually. So it's as a result of the secretion. So hyperthyroidism is a result from the secretion of the thyroid hormone. And if I ask you, what is the example actually of the hyperactivity of the thyroid hormone? This is Graves' disease. And again, Graves' disease is an immune system actually malfunction. In this disorder, our immune system produces some autoantibodies that bind and activate the thyroid stimulating receptors and leading the continual stimulation of the thyroid hormone synthesis. Because we have the axis control the thyroid hormones. TRH from hypothalamus and TSH from pituitary gland, and finally they produce actually T3 and T4. Whenever your body produces this hormone, you can stimulate the thyroid glands to produce some more hormone. Sometimes, as you can see here, as a result of the presence of autoantibodies. What is autoantibodies?
It's not in control.
We have antibodies and autoantibodies. When we say autoantibodies, what does it mean? It's a general concept.
Are they antibodies which are for the attached to structure within the body, like natural structures?
You know, sometimes our immune system produces some antibodies against the normal molecule of the body by mistake, okay? And many inflammatory diseases, like arthritis, like for example Graves, like Hashimoto, they are as a result of the production of antibody against the normal molecule. For example, TPO is a normal enzyme. Sometimes my body produces the antibodies by mistake, okay? And for example, about the arthritis, actually our body produces some antibodies. They can actually affect the joints, okay, the bone, the muscle, the function of the muscles, okay? So whenever, for example, in case of Graves' disease, we have some autoantibodies. For example, this is the receptor for the function of the TSH. And in the Graves' disease, autoantibodies bind and activate the TSH receptors. For example, normally TSH bind to these receptors. For example, these are not antibodies, okay? These are receptors on the surface of the cells. And sometimes my body produces for example, this is the receptor, and my body produces some antibodies that they mimic the function of the TSH, okay? This antibody mimics the function of the TSH, so they send the signal to the thyroid hormone to produce more T4, T3, and T4 hormones. Clear? So this is the actual mechanism of the occurrence of the Graves' disease. And as I mentioned, the symptoms and the signs are opposite of the hypothyroidism. For example, they are also all the time nervous. They have a heart rate they have a tachycardia actually. They have some problem in eye, and also all the time they are anxiety. About the Graves' disease is commonly actually treated by some anti-thyroid drugs. For example, one of the most important is the methimazole. Okay, so in a case of hypothyroidism, you should prescribe a thyroid hormone like a levothyroxine. So in a case of hyperactivity, you should prescribe some drug to suppress, okay, to suppress the activity of the thyroid gland. One of them, for example, is nowadays very common, is the methimazole. About the regulation, as I mentioned, we have one axis that starts from hypothalamus and pituitary and also thyroid gland. The production of thyroid hormones, I mean T3 and T4, is primarily regulated by the TSH. You should know that which hormone actually produced from which organ, okay, which part of the body. You should know that TSH originated from anterior pituitary, okay? And not only we have a TSH, in the actually higher level, we have a hypothalamus. Hypothalamus actually produces some releasing hormone, okay? They can control the activity of the actually many functions. One of them is controlling the activity of the thyroid hormone. And most of them contain R. For example, what does it mean? TRH, thyroid releasing hormone, okay? So most of the hormones that originated from hypothalamus, they are releasing hormones. This means that they can stimulate the release of some hormones. In the case of thyroid, they can stimulate the release of TSH, or thyroid stimulating hormone. TSH release in turn is stimulated by the thyrotropin. Thyrotropin means TRH, or actually TRH in an abbreviation, and it can release in a pulsatile manner. You should know that we have some feedback inhibition. As you can see here, we have a feedback inhibition of the thyroid hormones TSH and also TRH. What does it mean? It means that whenever our thyroid hormones produce actually a high amount of T3 and T4, it sends the signal not only to the pituitary but also to the hypothalamus to inhibit the release of TRH and TSH. So this process is known as actually feedback inhibition. So, actually, because as I mentioned, today we have some immunoassay tests, and one of them is the ELISA. And I mentioned to your friends, we are waiting for the factory to set up the printer of the ELISA reader. So inshallah, for the next semester, you have a ELISA in practical, okay, for the beta HCG. But I want to just review what is the principle and what is the concept of the ELISA. Have you ever heard this name ELISA? Do you have any information about the ELISA? No? Okay. ELISA actually is one of the important kind of the immunoassay. Immunoassay means that in this type of technique, you measure the concentration of unknown analytes by the formation of the immune complex. What does it mean, immune complex? It's a combination of antigen and antibody, okay? But this is a specific. So, immunoassay is a biochemical test that measures the presence of the concentration of the, most of the time maybe it is small, and sometimes you have a large molecule like a beta HCG for detection of the pregnancy, okay, or the age of the fetus. And you should know that this concept, when we say analyte, what does it mean? Analyte is the molecule that detected by this technique. For example, if you measure the concentration of T3, analyte is T3. If you measure the concentration of beta HCG, the analyte is beta HCG. If you want to measure the concentration of estradiol, testosterone, or HIV antigen, okay, for the next session you measure the actually HIV virus antigen. So this molecule known as the analyte. This is their one definition. And analyte may be present in some liquids. You should know that you should prepare it, the desired actually molecule in a liquid. Maybe you want to use a urine, especially for example, for the pregnancy strip test. Okay, home pregnancy test, you use a urine. And also maybe you want to use a whole blood. Maybe you want to use for example, serum or the plasma. Salaam alaikum. Too late. But you should know that one of the important parts of the ELISA actually is the antibodies. I think you're familiar with the structure of the antibodies. Am I right? What's antibodies?
They bind to foreign cells so that the antigens on the white blood cells can recognize it.
Mm-hmm. So, and I think you're familiar with the structure of antibodies. Am I right? This is the general structure of the antibodies. And we have actually constant domain and we have a variable domain. So this variable domain known as the antigen binding site. And this site is specific for any kind of the antigens. Maybe it's specific for the T3, beta HCG, I don't know, estradiol, testosterone. Any molecules are specific for this site. And we have a different type, IgG-A, immunoglobulin A, immunoglobulin D. So we have a different type. What about ELISA? Okay, please do not write ELISA Z actually. Okay, this is S. ELISA is the abbreviation of enzyme-linked immunosorbent assay. ELISA actually, or in general thing, immunoassay can actually present in many different forms. This is direct, indirect, or competitive and non-competitive. As I mentioned, we have the immunosorbent assay. Immunosorbent means that you have one plate, okay? Actually these are, first to show you these plates. These one, actually, these are the plates of the ELISA, okay? So, for example, in a laboratory, you take a test tube, okay, and you add a reagent, you add your analytes, okay, for example, glucose, sugar, amino acid, sugar, serum, urine, okay, do you remember from the previous semester? So, what in an ELISA, you have a specialized test tube, and these are actually known as a plates, or because the size is small, also known as a microplates. And each part are known as a well, okay? This actually each part known as a well. And you should add the reagent to each well, okay? So this is actually general container of the ELISA. As I mentioned, this is the immunosorbent assay. This is the immunosorbent assay. For example, assume that this gray plate is one of the well, okay? And I want to show you what happened to each well. So this plate actually considered as the one well of the microplate. At first, depending on the type of ELISA, you have a different form. For example, whenever you have direct ELISA, what does it mean? You have some antigens that coated. Coated means that immobilized. Okay? Then you take a plate at first. You add the antigen and immobilize, it means that fixed. Okay? Then you fix the antigen in a general and scientific name in ELISA known as a coat. Then you add antigen and fix it known as a coat. Okay? So, for example, you coat one antigen here. Maybe this is, for example, beta HCG. Maybe this is the T3. Okay? You fix it. And after that, you add a labeled enzyme here. Actual labeled antibody. This is the antibody that is against this antigen. Can we see the formation of the immune complex? For example, please consider that you don't have any enzyme here. You have only antigen and enzyme. Can I see the result? No, it is not detectable. So you should add the one label, and in ELISA, the label is enzyme. So if I ask you, what is the label in the ELISA? This is an enzyme. Sometimes, for example, in the next session, you have some dye, like for example, gold. You have a latex particle, they produce a color, okay? But if I ask you, what is the kind of label in ELISA? As the name implies, it's an enzyme, okay? And most of the important enzyme in ELISA actually is HRP. HRP is the horse radish peroxidase. Horse radish because many years ago did extract from horseradish. Okay, so now at the HRP. So if I ask you what is the label, maybe it's label to the antibodies, maybe in another test I will show you, maybe this is the antigen, okay? Just know that in ELISA, the label is HRP. This is the kind of enzyme, okay? So whenever you add antibodies that's labeled with enzyme, so enzyme, only enzyme actually, if you have enzyme, can produce a color? No. You should add a substrate. What chromogen? What does it mean chromogen? It means that after converting the substrate to the product, they produce a color as well. So this is the type of direct. In a case of direct, you should know that you should consider the immune complex. For example, this is the antigen, and this is the antibody. So, what is the main immune complex? I want to show you how we can distinguish between direct and indirect. Whenever you have the immune complex, this is the immune complex, this is the antigen, this is the antibody, and totally these are immune complex. You should consider these points whenever the label, I mean the enzyme, is directly bind to the immune complex. It is known as a direct ELISA. But what about, this is a presentation, actually I draw this one. Consume that, for example, this is a well. Not only antigen coated, sometimes you have some antibodies that coated, okay? Maybe in this case, you have a coated antigens. Sometimes in some type of the actually ELISA, you according to the requirement, maybe you have to put antibodies, okay? And after that, you added antigens, okay? And maybe this antigen is bind to the label enzyme, okay? No matter you coat the antigen or you coat the antibodies, whenever the label directly bind to the immune complex, this is the antigen antibody, okay? Whenever it's directly bind, now as a direct. So, for example, and you block the empty sites for the inhibition of the non-specific binding, and you add, for example, antigen that bind to the enzyme. By washing, you remove the unbound. And after adding substrate, the color change, and especially for the next semester, because you have ELISA finally, after adding substrate and you stop your enzymatic reaction, you have a yellow color, exactly like this, okay? And you can read the absorbance. Okay, what about indirect? Who can describe the difference according to this shape? Difference between direct and indirect.
In direct, the antibody bounds the antigen, and then the enzyme binds it to that direct. But it seems here that the antibody has another antibody bound to it, and then that has the enzyme.
As your friend explained, as you can see here, in a direct, actually, in the direct, as you can see here, the label binds directly to the immune complex. What about indirect? What is the immune complex? This antigen and this antibody. So this part known as a desired immune complex. But as you can see here, the label is actually a part from the main immune complex. So the blue antibody is important for us, okay? As you can see here, because the label is out of, for example, if I want to draw it here, you have a coated antigens. And you have a first antibody. Can you see any label here? No. But what is the main complex for us? This one. Okay? This one is the main complex. Can you see any label like this? No. So this kind known as a direct. But whenever you add a secondary antibody or another antibody, and that antibody binds to the enzyme, because this label is apart from the main complex, it is known as a indirect. Okay, clear the difference?
Yes.
And for example, this is another, you coated the antibody, you block the empty site, and also, for example, these are the antigens that bind to the other molecule. This is the biochemistry metabolism. It's not necessary to know the details, just know that sometimes you don't have any secondary antibodies. We have a second actually enzyme because this complex known as the main complex enzyme is apart from the main complex, also known as an indirect ELISA. Another classification, no matter this is the direct or indirect, is a competitive actually or non-competitive. When we say competitive, as the name implies, you have some competition for the binding of the antibodies. So, for example, consider this point. In a one well, okay, in a microplate, and you added, for example, serum, and you added one molecule that is very, very similar to a molecule. For example, in a T3, if you have an ELISA test for the T3, because T3 hormone, as I mentioned, is very small. You know, you have a different type of ELISA. For example, maybe you want to measure the concentration of beta-HCG. Beta-HCG, what is it? It's a large protein, okay? But depends on the size of molecules. And for example, this is the T3. The size is very small, okay? As I mentioned, this is the tyrosine that binds to the 3-iodine. As you can see here, the size of the T3 is very small. So the design of the ELISA is different. At first, I'm going to talk about this small molecule. No matter, maybe you want to measure the concentration of morphine for the average person. Okay, it's very common to measure the concentration of some drugs like morphine in the blood, in the urine. Okay, because they are very small. If they want to measure the concentration of morphine in the urine or in the blood, we require to have one specific type of ELISA. For example, at first, in a one well, okay, one of the well of microtiter, you have coated antibodies. For example, these antibodies are against T3 hormone, okay? And after blocking the empty site, you block it, for example, by the specific proteins. You added, for example, serum of your patient, okay? And for example, you want to measure the concentration of T3. Whenever you add a serum, these are T3 hormones, okay? At the same time, you add one reagent. It's ready to use. What is reagent? that labeled with the HRP or enzyme is very similar to the T3. When we say competitive, it means that you have a competition with two similar molecules. You know, if I have this kind of molecule, is it correct or not?
No.
Because the shape is different.
Yes.
For example, you have a match and you want to evaluate the speed of the person. For example, about my case and another person with a 150. It is not actually correct to have a match and competitive, okay? So, whenever you have a competition, the shape should be very similar. So, whenever you add a T3 and this label, they compete to each other for binding to the antibodies. Please tell me when we can see color in the well and when we cannot see color in the well. Please tell me according to which fact you have a competition, as I mentioned, you add a zero, okay? You add a zero and also you add a, for example, label together at the same time. Please tell me whenever this one binds, whenever this one.
They are at the same time.
At the same time,
At the
okay?
same time binding to the receptor. So like when we have like 50 receptors, in the competitive 25 of T3 binding, and the other hand, the 25 they also bind to receptors. So I measure the density of color of this 25. Then I can see the concentration because I add 50 receptors. Then I have the color for 25. So the 25, the other 25 is binding to T3.
I want to ask you this question. In a case of hyperthyroidism, what does it mean, hyper? It means that you have a high T3.
Yes.
So, what would be the color here?
It's less than.
Ahsan. Because in a case of hyperthyroidism, in a competition between serum and this label, which one wins? Which one wins?
Serum.
T3 wins. It doesn't allow to the HRV label to bind here. So can you see any color?
No.
No. So what happened in a case of hypothyroidism?
We see color because the T3 is low.
Because T3 is very low. Finally, in this match, because in a hypo the concentration is very low. So only these labeled bind to the actually substrate. So I want to describe the next slide for you. So if I draw a curve, because in ELISA finally after adding substrate it produces the color, yellow color, and you read the optical density. Okay, do you remember the spectrophotometer? You read the absorbance. ELISA is very similar for the spectrophotometer. Why? Because finally you read the absorbance of the colored product. So if I want to draw the concentration, if I want to draw a concentration against OD, please tell me, in a case of hyperthyroidism, for example, 100 millimolar, for example, what would be the OD? Aspen. It is low. In a case of low concentration,
So we have
Aspen. So you have a descending line. Clear? Yes?
yes.
So this is the actually curve of dose response. As you can see here, whenever the concentration is high, they don't allow for the bind of nasal enzyme. So the color is very low because we have a low enzyme here that can produce a color. But in a case of low concentration, like for example this one, most of the conjugated enzyme actually they bind here and they convert the substrate to the product. So you have a high OD, okay? Clear? So you have a high OD. So if I ask you about the definition, you should know that which of the, for example, I ask you which of the following sentence is correct about the competitive. Maybe I ask you, in a competitive metabolic, the dose response curve is directly proportional to the concentration. Clear? Correct?
No.
No. Because they have actually we have a descending curve, or there is an inverse, okay? There is an inverse relation between concentration and OD. In other words, when the concentration is low, OD is high. When the concentration is high, OD is low. Okay, there is an indirect relation between OD and concentration. Okay, what about the non-competitive? As the name implies, there is no competition here. What is it? This is especially for the large molecule. Like which molecule I described for you? HCG. Okay? This is the hormones that evaluate for the pregnancy. If it is positive, it means that your patient is pregnant. Okay? And also you follow the pregnancy. And you can see here, because the size of the molecule is very large, we cannot design as a competitive, okay? Only a small antigens, small analytes, like morphine, like a T3, like a T4, they can design for the competitive. What about large molecule? You can make a sandwich, okay? Maybe you can make a sandwich, okay? So, what is here? For example, you have immobilized or coated first antibodies. This is the antibody number 1. And you have a large beta HCG, for example, this is the beta HCG as the antigen. You can make a sandwich for this large molecule. And you have a label here, enzyme, and following the adding of the substrate, the color of the substrate changed to the yellow, and you can read the absorbance or optical density. So please tell me, this type I draw for you, it is a direct or indirect? Why indirect? Direct. You should know that the main immune complex is this one. Okay? Whenever you have this one, actually, it is indirect because the enzyme is out of the main complex. Okay? So, as you can see here, this is actually immunoassay that there is no competition. So, if I want to draw the curve, what is it whenever the concentration is high? What is OD?
High.
High. The concentration where it is low, it is that. So this is a direct relation between concentration and OD. This is the type of sandwich because they make a sandwich between target, especially target proteins, large molecule. It is often known as a non-competitive. As I mentioned, maybe sandwich assay may be present in a direct first antibody, large protein, second antibody, and directly enzyme. About indirect first antibody, molecule of proteins, second antibody, and out of this immune complex you have an enzyme labeled. So as I mentioned, for the next semester, inshallah, you will do this procedure. At first, you should take a specialized test tube, and the name is a microplate. Okay, they have actually specific characteristics. They made from polystyrene. They are special for the ELISA test. And in a case of after, for example, adding antigens, antibodies, and enzyme, you add a substrate and you stop your solution. Finally, you have a yellow color. How can I read the absorbance? By the specific type of spectrophotometer. The name is ELISA reader. These are, I think you have this type. This is the American type of ELISA reader. Okay, so as I mentioned to your friends, we are waiting for the setting of the printer for this instrument. And inshallah, for the next semester, you will have an experimental about the ELISA, I think for the beta stage. So this is a microplate reader. As I mentioned, it's very similar to the spectrophotometer. And instead of cuvette, you remember, you add a sample solution in a cuvette and read in a spectrophotometer. So instead of cuvette, you have a well. Each actually well considered as a cuvette. And what is the difference? Maybe I ask you the difference for your final exam. Maybe I ask you about the difference between a spectrophotometer and ELISA reader. You should know that the path of light transition is vertical, okay? If you remember about the spectrophotometer, we have a lamp and the path of light was horizontal. About this spectrophotometer, actually this is vertical position. And they are specific for the, they have a very specific filters for the different wavelengths and smaller volume. For example, for the spectrophotometer, at least you require 500 microliter of half milliliter. Do you remember? This, you work with a sampler, 500 microliter or lambda, okay? But in the case of ELISA reader, maybe 200 or maybe 100, okay? Lambda or micro, okay? These are not milli. Microliter can read by them, actually ELISA. And also whenever you bought ELISA kits, they contain some standard solution, they have some microplates. And I didn't describe the next slide because we don't have a practical session. I want to just describe how we can determine the standard and unknown sample. As I mentioned, you have a different standard solution. And you should know that this kit, for example, for the T3, is actually contain a special concentration of the T3 standard. If I want, please tell me, if I want to draw a standard curve means dose response curve about the concentration of the T3 and about OD. Please tell me, this is a T3, so this is a competitive ELISA. If I want to draw a standard curve or dose response, standard curve. If I want to draw a standard curve, this is the ascending or descending?
Descending.
Excellent. So at first you take a different solution and following do this procedure. For example, you have a 1, 2, 3, 4, 5, 6, 7, 8, and 9. For example, nanomolar, okay? And after adding, because, for example, this concentration is high, it doesn't allow the binding on conjugated, so the OD is low. And by this one, we find the OD, so you have a descending. So you have actually a standard curve. And for the next semester, actually maybe your professor gives you an unknown serum, okay? And she already asked you determine the concentration. How we can do this? After proceeding the ELISA, finally you have an OD, for example like this one, maybe 0.5. So what is concentration? You should find the intercept and find the x-axis intercept. So these points now as a concentration of unknown sample. Clear? So according to the standard curve, at first you should draw a standard, maybe she or he asked you to draw it in Excel, okay? And finally you have a line and you read the absorbance of your, for example, sample number 1, sample number 2. And according to the OD, you find the intercept to the line and find the intercept to the x-axis. So because x-axis is concentration, finally you can see the, for example, maybe this is the 5.5 nanomolar. Clear? This is the basis of the ELISA. Swal?
No.
No.
Okay. I think when you want to start the next session?
15, 20 minutes. Or 20 minutes.
Okay, 10 actually.
Oh.
Inshallah, we'll start it. So, for the new coming students, would you please come one by one, check your name,